Thank you for hosting us. We're glad that we can attend the presentation here again after last year. And we've made quite a progress since last year. We filed a 510(k) for our branch point device, and we also licensed a new patent that covers the chemotherapy local delivery of 2 drugs for the treatment of brain tumors. And we also raised a $2 million funding in June. So let me talk a little bit about our company to give you an overview of us. So we're a biotech company focused on the development of novel neurological therapies to be directly delivered to the human brain. And we have a development pipeline that's based on the clinical science from the University of California in San Francisco and Johns Hopkins University. Our first program is the branch point device, which we licensed actually last year when we spoke here at the conference, which allows the local delivery of a number of therapeutics, including stem cells, but also gene therapy or chemotherapy to the human brain. And the development of the device was originally funded with almost $2 million by the California Stem Cell Agency. And we filed a 510(k) in June of this year. Our second program is the ACX31 program for the local delivery of 2 chemotherapy drugs for the treatment of brain tumors. Those drugs are temozolomide and BCNU or carmustine. And here's an overview of our pipeline. Our branch point device, as I said, is in 510(k) review by the FDA. Our ACX31 program is earlier stage in preclinical. However, we believe that there's potential for a shorter 505(b)(2) pathway, given that the underlying molecules are already approved by the FDA. So let me talk about the branch point device, which we introduced last year. So if you look at current standard of care, right now, most of the time, a straight needle is used for the delivery of stem cells. The issue with that approach is that if you have to cover a certain area in the brain, you often have to penetrate the brain multiple times just because the needle is straight and you have to kind of work around that area. So some patients with Parkinson's, for example, they received up to 16 separate brain penetration to receive their stem cells. Our device, the way it's designed is that you have a single penetration with a guide catheter, which is the picture in the middle. And then within the guide catheter, you have a delivery catheter that's curved and flexible. This allows you to penetrate or deliver stem cells horizontally and into different areas in order to target your entire target area with only a single penetration. So this also allows you to avoid these multiple penetration that you see with a single steel needle, which can cause tissue damage, stroke, and bleeding, of course. Also, another advantage of our approach is that you can avoid the problem of reflux of the infusion volume. If you look at the bottom pictures, that is a straight needle, and when you infuse that, you actually see how the infusion volume actually comes out to the surface through a reflux along the injection channel of the steel needle. With our device, given that it's curved, if you pull it back, you actually see how the contrast agent in this case actually stays in the target area, and you don't see reflux. And we also use that technology, our branch point device, to deliver stem cells to human pig brain as a proof of concept, and we were successful to do that. And those stem cells, those are human dopaminergic stem cells, actually were able to survive in the pig brain through the delivery with our branch point device. Then our second program is our ACX31 program to deliver chemotherapy locally to brain tumors. This is a picture of gliadel, which you might be familiar with. That is actually a local chemotherapy wafer to deliver one of the drugs that's covered by our patent, which is BCNU or carmustine. So typically, after resection of the tumor, you have a resection cavity, and the surgeon places those chemotherapy wafer into that resection cavity in order to treat the margins of the tumors that are not removed. So the patent that we licensed from Johns Hopkins covers the local delivery of temozolomide and BCNU, and we licensed that from Dr. Brem, who is the Director of Neurosurgery at Hopkins. And our goal is to deliver that either either as a wafer formulation similar to Gliadel, or with a microparticle formulation that we can inject with our branch point device. And the temozolomide itself as an oral tablet used to be a very significant product or drug. It used to have $400 million in sales in the US, almost $1 billion worldwide as an oral tablet under the brand name Temodar before it went generic in 2012, 3 years ago. So if you look at the current standard of care of treatment of glioblastoma brain cancer, oral temozolomide is being used as first-line treatment. Almost all patients receive oral Temodar today. The drug was approved with a survival benefit of 2.5 months. Nonetheless, the drug was commercially pretty successful. And Gliadel had a similar survival benefit of 2.3 months. The third drug that's being used today for treatment of glioblastoma is Genentech's or Roche's Avastin, not a chemotherapy drug. But if you look at local delivery of chemotherapy, first line, second line today, our patent covers that. And that also creates a relatively large market potential for local delivery of chemotherapy to treat glioblastoma, just by the fact that temozolomide or Temodar went generic, creating a relatively large drop-off in sales that we believe we can potentially recapture with our product. Here again is an overview of the survival benefits of oral temozolomide and Gliadel, or local BCNU. Relatively modest survival benefits of a few months compared to just radiation therapy that typically allows you to survive 12 months. The issues here is really the blood-brain barrier. If you deliver a chemotherapy drug through oral administration, typically 10% or even less of the drug actually ends up in the brain tumor. However, you have 100% of the side effects. So our goal is to circumvent the blood-brain barrier through local delivery. And if you look at the cost of treatment, when Temodar was a brand, we're talking about $30,000 or $40,000 per patient, which is fairly consistent. And if you can improve survival beyond the 2 to 3 or maybe 4 months that you see with current standard of care, there could be even more potential to charge a premium price for that. And here's what Dr. Brem at Johns Hopkins did with local delivery of temozolomide. He used oral delivery of temozolomide in rats to see how long those rats survived. It was about 20 days. These are rats with glioblastoma or brain cancer cells. And the fact that temozolomide was locally delivered actually increased the drug concentration threefold, and they saw a significant increase in survival benefit. This is our team right now, so you'll see we'll cover a number of, we have experience in a number of previous medical device and also pharma companies. And if you look at our clinical and scientific advisory team, again, as I said, Dr. Brem at Hopkins, Bob Langer and Dr. Brem, they actually work together in terms of developing the Gliadel formulation, and both are advising us now and when we're collaborating with them to develop a extended release formulation that can locally deliver not only temozolomide but also BCNU. And we're also working with Betty Tyler, who works with Dr. Brem, and Daniel Lim, who invented our branch point device at UCSF. He's working with us as well. So let me conclude with a financial summary of our company. So we're publicly listed on the OTCQB under ticker symbol ACXA. We have approximately a $10 million market cap. Our shares outstanding as of August was 6.3 million shares, and We raised $2.25 million in June. The lead investor was Savvy Capital, and as of last quarter, we had $2.3 million in cash. Thank you.